Assalamu alaikum and welcome to the 3rd grade 12 biology Telikilaas. In today's class, we are going to be discussing about the stages of photosynthesis. But before we start, let's recall some of the things you learned in the last class regarding the photosynthetic pigments. So I have here some of those terms we covered in our last class, just 4 of them. What I want you to do is I'm going to be showing you 5 different pictures. 4 of them will be matching with these terms, and I want you to identify which of these pictures match correctly with these terms. You have 15 seconds to do so. Your time starts now. Alright, your time is up. Let's see if you're correct. Were you? Let's discuss these terms. We'll start from the bottom right over here. So this 5th picture shows you the results of a chromatography, and you're asked to identify what this number 0.43 refers to. So you can see from this 5th picture that it is really the ratio of how much this second pigment has moved from the baseline compared to that of the solvent. The term that we use for such ratios is called an RF value. And you know that RF values can be used to identify what these pigments are. So we could use it to identify what this second pigment is. And we also learned in the last class that these pigments that are in plants, they can absorb different wavelengths of light. And the graph that shows how much of light is being absorbed at the different wavelengths by these different pigments, it's referred to as an absorption spectrum, right? So that's this 4th picture. A similar graph is the action spectrum shown in this 3rd picture, which is the graph that shows how much of photosynthesis occurs in these different wavelengths, right? And we also looked at one of those pigments, the structure of chlorophyll, and we learned that in chlorophyll it has a porphyrin ring and it also has a phytol tail. Now in these 2 pictures at the top, although both have a porphyrin ring, only the first one is chlorophyll because it's the one that has magnesium as its central atom. Okay, and in this picture, although it doesn't show the tail itself, I just have it in a condensed form. So if I were to show it in a displayed form, this is going to be the hydrocarbon phytol tail and this is going to be the porphyrin ring. And the second molecule, the second picture, is really a heme group. So it does have a porphyrin ring to it, but instead of a magnesium, it has a central iron atom to it. And it's this component, this heme group, that's going to be embedded within the protein part to form the hemoglobin, which is the one that's carrying oxygen around your body. Now let's talk more about how light is captured, and also we discussed about how chlorophyll can absorb the energy from light in the last class, and we also discussed in the previous class that the light energy being captured by chlorophyll allows for the photosynthetic reaction to occur. And I told you then that photosynthesis doesn't occur as a single step, it's a many-step reaction and there are quite a few details to it. So today what we're doing is we're going to be looking at what are these different stages of photosynthesis, okay? And as we do, we're going to focus the lesson on these objectives. So by the end of today's class, you should be able to explain the role of the photosynthetic pigments in photosynthesis. You should be able to state what are the main stages of photosynthesis, and you should also be able to identify where these stages occur and what are the major components involved in these stages. You're also expected to be able to define this term photophosphorylation as well. So let's get started. Now because you know that photosynthesis is the process where light energy is captured to synthesize the sugars, you would have realized that the capture of light energy should be definitely one of those first steps in photosynthesis, and the production of sugar should be one of those last stages in photosynthesis. Okay, but before we actually look at those stages involved, we'll first have a look at how this light energy is harvested, because it's definitely one of those first things that's going to happen, and because it's related to our previous lesson too somewhat. Now looking at just this equation here, you might think that even a couple of chlorophyll molecules could be enough to carry out this process, but one chlorophyll molecule or even 2 cannot capture enough light energy to synthesize the sugars to carry out photosynthesis. So chlorophyll molecules, they will work in groups of about 100 or so different pigments, all working together, and these pigments, they are going to be embedded in a protein complex that's going to be found in the thylakoid membrane. So this protein and pigment complex, it's called as the photosystem, and that's where you find the chlorophyll molecules. Okay, and of these different pigments, there's always going to be one pigment that's slightly different from the others, a specific chlorophyll A molecule in every photosystem, which is the reaction center. Okay, so every photosystem is going to have a reaction center, and it's also going to have these other molecules which are referred to as the accessory pigments. So you have the reaction center and the accessory pigments. Now, whether it's the reaction center or whether it's part of the accessory pigments, their purpose would be basically to absorb light energy, but what it does after it absorbs the energy is what's different about it. For example, the purpose of the reaction center would be, after absorbing the energy, to excite electrons. So when it excites these electrons, the energy in them would be used to synthesize other molecules, which through a series of steps will be ultimately producing the sugars. Alright, but a reaction center on its own cannot absorb enough light energy, so it's going to need all of these accessory pigments. And those accessory pigments, because you have those carotenoids, xanthophylls, the chlorophylls and all, each being able to absorb different wavelengths of light, that energy in those different wavelengths can be harvested, and each of those accessory pigments, they can really funnel this energy to the reaction center, so that the reaction center can excite the electrons, and so that the photosynthetic process can be initiated. Alright, now looking at the actual stages here, so I told you already that it's going to be the harvesting of light should definitely be in the first steps. So if you look at the stages of photosynthesis, photosynthesis occurs in two main steps. The first is going to be the light dependent reactions, and the second is the light independent reactions. And because light energy is going to be captured in this first step, the first major step, it's called the light dependent reaction. So light dependent reactions occur in these membranous structures, which you would remember are called the thylakoids, whereas the light independent reaction occurs in the stroma of the chloroplast, which is this fluid part of the chloroplast. The light independent reaction is also called as the Kelvin cycle too. So if you look at the light reactions or the light dependent reactions, this is where the light energy is absorbed. This is where the water acts as the reactant, and it's also here that the oxygen is going to be produced. Whereas in the light independent reaction is where carbon dioxide is the reactant and glucose is the product. But if you look at these equations here, you can see that they are not quite balanced. The numbers of the elements on either sides don't match up, which tells you, for example, if you look at this one here, you can see that although hydrogen is found as part of the reactants here, it's not found as any of the products here, right? So the same occurs in the light independent reactions as well. Although hydrogen is here in the in the products part of it, it's not there as one of the reactants involved, which tells you that the hydrogen is definitely being transferred to another molecule, right? So there are other molecules involved in this light dependent and the light independent reactions, and these are going to be the NADP plus ADP, ATP and so on. So these are the molecules that we are going to be looking at a little bit in more detail in today's class. Now, so if you look at the light dependent reactions again, in addition to the water and the light, you also do have the NADP plus the ADP in the inorganic phosphate as the reactants, and these together is going to form the oxygen gas and the ATP and the NADPH. So what I want you to do now is, if you haven't taken out your notebook already, please take it out, and I want you to complete these equations to show what are the reactants and the products for the light dependent reaction as well as the light independent reaction by using the information on this diagram. You don't have to balance the numbers on each side. Just use the information in this diagram to complete these equations. Okay, I'll give you some time and then we'll discuss the answer. Alright, I hope you've done. So let's check your answers. So if you look at the light dependent reactions, you can see that in addition to the water and the light, we have NADP plus ADP and inorganic phosphate as the reactants of it. And then the products, in addition to the oxygen, we also have ATP and something called an NADPH that is being produced, right? And this ATP and the NADPH, they become the reactants for the light independent reactions, and it's during this process you have the glucose that's going to be produced, right? And in addition to the glucose, during the light independent reactions, the NADP plus ADP and inorganic phosphate is going to be reformed so that these can be used again as the reactants for the light dependent reactions. So you can see basically this ADP and ADPH and ADP plus and stuff, these are basically being recycled within the light dependent and the light independent reactions, right? Now let's also look at the structures of these molecules a little bit more detail and talk about what it does to some extent, because in later classes we are going to go into the details of this light dependent and the light independent reactions. Okay, so we'll start with the ATP molecule because that's the one you're going to be more familiar with. So remember what ATP stands for to start with. Well, ATP stands for adenosine triphosphate, right? And adenosine triphosphate is basically a modified nucleotide. And what are the components of a nucleotide? It's going to have a pentose sugar connected to a nitrogenous base. The pentose sugar is also going to be connected to a phosphate group, right? So that's the basic structure of a nucleotide. Now, when it's adenosine triphosphate, it will have specifically a ribose sugar and an adenine base. So it's the adenine and the ribose together that makes the adenosine part of it. But if it had just the one phosphate, it's going to be called adenosine monophosphate. So in this case, since it's triphosphate, how many should there be? There should be 3 phosphate groups, right? So adenosine triphosphate, if you remember the function of it at all, would be the molecule that is which acts as the immediate source of energy in the cells. And to supply this energy for the cells, what it has to do is it's going to break these covalent bonds that are connecting these phosphate groups together. So to do that, what it will need is an enzyme and water. So when water is added across this covalent bond by this enzyme ATPase, it's going to release the energy in this bond to form adenosine diphosphate and an inorganic phosphate. Now this is called an inorganic phosphate because the phosphate group here is not going to be attached, it's not going to be involved with any carbons and hydrogens like in the organic molecules. So hydrogen, the water is going to be added across this covalent bond to break the ATP to ADP and inorganic phosphate. And because water is the one that's involved in breaking this molecule, it's called a hydrolysis reaction. And in this exothermic reaction, the energy that is going to be released is going to be enough to supply for the different chemical reactions, just one step at a time. And it's this energy that's going to be used to carry out the anabolic reactions, meaning for every one of those steps where molecules are being synthesized, larger molecules are being synthesized, it's ATP, the energy in ATP, that's going to be providing the energy for it to be synthesized. So synthesis of sugars, the proteins, the DNA all require ATP to be present and the energy to be released by hydrolyzing the ATP. In addition to this, ATP hydrolysis, the energy from it will also be used for the cell division. It's what's going to be used for active transport. In animals, it's also going to use this energy for muscle contraction, for maintaining the body temperature, and even impulse transmission. And energy, when available, can actually be used again to combine these two together to reform the ATP molecule and the water. And because water is going to be released in this process, this is called as a condensation reaction. This is specifically a phosphorylation process because you have a phosphate group being added to the ADP here. Okay, and the energy, and this is going to be done by an enzyme, ATP synthase, or sometimes even called ATP synthetase. So you can fairly easy guess from the name itself, this is the enzyme involved in ATP synthesis, right? And this energy for this process is going to be in either of two forms. It can, in the case of respiration, it's the breakdown of glucose molecules. It's going to provide the energy for the synthesis of ATP. Whereas in the case of photosynthesis, it's the energy from sunlight that was captured that would be used to synthesize the ATP. So in photosynthesis, the energy in light after being captured is going to be used to synthesize the ATP, and the ATP that is synthesized would be used later on to synthesize the sugars. Okay, and when ATP is synthesized in the presence of light, there's a specific name for it. So that is what photophosphorylation is. So photophosphorylation, the photo refers to the light part, phosphorylation means to add the phosphate group. So photophosphorylation would be the production of ATP in the presence of light, specifically in photosynthesis. Okay, so if you look at the previous equation that we had, you can see that in the light dependent reactions, the energy from light is really used to synthesize ATP, and this ATP is what's used to, the energy in the ATP is what would be used to synthesize the sugars. Alright, now let's also look at this other molecule, NADPH. Looking at this here, you can see that in the light dependent reactions, NADP plus accepts these hydrogens from water, and that hydrogens from water is what's going to be incorporated into the sugars later on in the light independent reactions, right? So let's look at what this molecule is and what it does and how it does it just a slight bit. So looking at the NADP molecule, NADP stands for nicotinamide adenine dinucleotide phosphate. Alright, now I realize it's a mouthful to say, so even if you simply say NADP, that is fine for you. But if you look at this term here, what do you think would be some of the things that it would have in it? So you know, phosphate is going to be one of those components. It's going to have adenine, so base should be there. And because it says dinucleotide, you would think that there is going to be two nucleotide parts to it as well. And that is all true. So if you were to look at the structure of NADP, this is basically how it's going to look like. Now, you don't have to worry that you are going to have to draw this because you will not be asked to do so. If you need it for any reason in the question, it will definitely be given in the question. Okay. But looking at the structure of this, you can see it does have quite a few similarities with that of a nucleotide. So at most what you could be asked about NADP, the structure of it, would be given a diagram of NADP. You could be asked about how similar or how different it is from a nucleotide. Okay. So looking at this structure from what we've discussed before, you would know that NADP acts in two forms. So it exists in the NADP plus form, the oxidized NADP form, and it also exists in the reduced NADP form, right? And what do you remember about those terms, oxidized and reduced? So oxidation is going to be, pretty sure you would have learned it as oil rig, right? So oxidation is loss of electrons, reduction is gain of electrons, right? So it's not just the electrons, but oxidation is also going to be the loss of hydrogen, the gain of oxygen, and the increase in oxidation number, whereas reduction is going to be the opposite of this process. So looking at these two diagrams, you try to identify which one is the NADP plus, the oxidized NADP molecule, and which one is the reduced NADP molecule, and try to guess why so. Okay, let's see. So on the left here is the oxidized NADP molecule. You know that this is the case because you see it does have this plus charge on it, whereas in this case you can see that it has electrons added to it, and it does have a hydrogen that was not in the previous case as well. So this molecule would be the reduced NADP molecule. So how do you go from the oxidized NADP form to the reduced NADP form? So looking at the structure, you can tell for reduction to occur, a hydrogen ion needs to be there, which is a proton has to be there, and 2 electrons need to be there as well, right? So the hydrogen ions are going to be donated from the water itself. The electrons are the ones that were excited by the reaction centers when light is being captured. Okay, so those when combined to the NADP plus molecule, it's going to reduce it so that it forms the reduced NADP molecule. And this reduced NADP can lose this hydrogen and the electrons to reform the oxidized form again. So in the light dependent reactions, NADP is going to get reduced, whereas in the light independent reactions it's going to get oxidized. So it's going to donate those hydrogens and electrons in the light independent reactions. Okay, so like that, NADPs, they act as electron carriers. They can transport these hydrogen ions and electrons from the thylakoid membrane where the light dependent reaction occurs to the stroma where the light independent reactions occur. Okay, so let's look at the equation once again. So in the light dependent reactions is where you have water split to form oxygen and the hydrogen in the water. You can see it's going to be donated to the NADP, oxidized NADP, to reduce the NADP molecule. And it's also in this process that the light energy is going to be captured and photophosphorylation occurs to form ATP molecules, right? And this ATP and NADPH is going to be used in the light independent reactions where the hydrogen from the water would now be combined with the carbon dioxide to form the sugars, and the energy from ATP would have been used to form the sugars too. And this is going to reform those NADP, ADP and inorganic phosphates, which can be used again as the reactants of the light dependent reactions again. So this equation, this definition that you had learned previously, that the photosynthesis is the process where plants will capture the energy in light to synthesize the sugars, can now, by using this information that we have learned, redefined to say that it's the process where energy in light is used to break apart these strong bonds in water so that the hydrogen can be stored in sugars by combining it with carbon dioxide, and that during this process oxygen is being produced and released into the atmosphere. Okay, and as you would know by now, we would have realized by now, the reason why in the overall reaction we don't have the ATP, the ADP, NADPH and NADP plus is because they are being, the same number of these molecules that are being used would be reformed later on, so they basically cancel out and therefore are not on the overall reaction here. Okay, let's summarize what we've learned so far in today's class. So we discussed about how in photosynthesis there are two main stages, right? There is the light dependent reactions and there is the light independent reactions, also called the Kelvin cycle. That in the light dependent reactions, light is going to be captured, and it's in this process water is going to be split and oxygen is going to be released. In that process ATP is going to be synthesized and NADPH is also going to be synthesized, which then are going to be used in the light dependent reaction, independent reactions, and that would be in that process where sugars is going to be produced, right? With that, let's see of the objectives how much we've covered though as well. So we've learned today about the role of the photosynthetic pigments. So the role of photosynthetic pigments would be to absorb the energy in light so that the energy can be converted into some other form, a chemical form, which can ultimately be converted into sugars, right? And the two main stages of photosynthesis would be the light dependent reactions and the independent reactions, where both the light dependent reaction occurs. So that occurs in the light dependent is going to occur in the thylakoid membranes, and the light independent reactions are going to occur in the stroma. And what are the major components of this? So the light dependent reactions will involve the light, the water, ADP inorganic phosphate and NADP plus, whereas the Kelvin cycle or the light independent reaction is going to involve the carbon dioxide, NADPH and ATP as the reactants, ADP inorganic phosphate and NADP plus, as well as sugar is going to be produced from the process. Okay, and what do you remember about photophosphorylation? So photophosphorylation would be the process whereby plants will synthesize ATP in the presence of light. So when ATP is produced in the presence of light, that's called photophosphorylation, right? So with these objectives being covered, we've actually covered the syllabus objectives from the syllabus document 5.1 and 5.2. Okay, so I hope this was useful for you. In our next class, we're going to be looking at the details of how the light dependent reactions occur. So how exactly is that ATP and NADPH are being formed? How exactly is that the light energy is used to synthesize this ATP? How exactly would photophosphorylation occur? Okay, so that's all for today's class. Hope to see you soon. Assalamu alaikum.